center talk. Please feel free to fill in at the front of the room here. Don't be afraid. Jim's a very nice guy. Also, even if you didn't sign up for a meal, we will have a reception and some dinner afterwards. So feel free to hang around after the talk. All right. So today I'd like to introduce our speaker, Jim Bever. Oh, see, you cannot rely on ChatGPT. This is not the American actor from Wyoming, nor is it our USAID Ghana mission director. Luckily, today we have Jim Bever. He's a distinguished professor of ecology and evolutionary biology now at the University of Kansas. And it's a special treat to have Jim here today. It is not often we have a new field of study invented, and we have the founder of that field here. Farmers have known for thousands of years that something happens in the soils when plants grow that changes how plants grow. But it wasn't really until Jim's work about 25 years ago that ecologists have had a way to understand how millions of soil organisms affect plant growth. And he's made two really key contributions. One was an experimental approach, a bioassay approach that lets us understand how all the organisms in the soil affect plant growth. And then he also developed a mathematical approach that helped us understand. And you can see here, it's a very complicated mathematical model. And so with these two advances that he's spearheaded, we've seen a really rapid growth in plant soil feedback research. This is a field of study that was just invented pretty much by Jim 25 years ago. And if we look at the number of papers on plant ecology versus plant soil feedbacks, it's been a tremendous rise, right? And if you extrapolate here, in 2040, we will all be doing plant soil feedback research. Jim is appropriately been recognized for his work. He's a fellow at AAAS, ESA, and Guggenheim. His about 300 papers have over 30,000 citations. His H-index is larger than the number of papers I've published. And on top of that, he is a super nice guy. He's always been extremely kind and generous with me. I have never heard anything but glowing reviews from his students and colleagues. So I would like him to know that you have a great impact, both personally and professionally, on colleagues around the world. And before I get all verklempt and start to cry, let me hand it over to Jim. Thank you, Andy. That was really nice.
So I've really enjoyed visiting with many of you. I'm super happy to be here. My son, I came to Logan, I think maybe 5 years ago, dropping off my son, who is a participant in Utah Conservation Corps. I'm not sure that program exists anymore. It was funded with the AmeriCorps fund. But anyway, he really enjoyed it. He liked living on the streets of Logan for 4 months and until it got really, really cold. So yeah, I'm going to talk about plant microbe interactions and how it can contribute to plant ecology, plant dynamics. And so today I'm going to talk mostly about pathogens and plant pathogen interactions. And then tomorrow I'll talk about mycorrhizal fungi, the beneficial side of things, mycorrhizal fungi. So I'm going to just start by introducing the not working. Wonder why. Orient you to where I'm going to be working. I'm going to be studying at the Tallgrass Prairie System. So this is an image of the National Tallgrass Prairie Preserve in Kansas. It's a bit fuzzy, but you can get an image of a fantastic landscape. And this ecosystem type covered from the Gulf of Mexico up into Canada, from central Kansas over into Indiana, actually patches in Ohio, Tennessee. So very wide geographic climatic region. And it extended into this high precipitation area because of a common management tool by fire for many, many native peoples that populated this area. So I'm going to study plant microbe ecology in the context of a system that co-evolved under native peoples management. So this system has many positive ecological attributes. It built tremendously fertile soil, sequestered tons of carbon in that soil, and by retaining the soil and resources on land, it allowed for clean waterways. Because it built tremendously fertile soil, it's been largely replaced by rural agriculture. And this system is qualitatively different. It's reduced soil fertility, burned off tons of soil carbon, and because it doesn't hold the soil or the resources in the soil on the land, it resulted in silted and polluted waterways in a massive dead zone in the Gulf of Mexico. So there are lots of things different about these two systems. I'm going to direct your attention to something not evident in these two pictures, and that is the difference in diversity. So the tall grass prairie has lots of long-lived perennial plants that co-occur stably over the long term. This is a prairie remnant near the Chicago region, while rural agriculture in this region is commonly just corn and alternated with soybeans. So this difference in plant diversity could have functional consequences. So here's results from an experiment in the tallgrass prairie system in Minnesota, where they manipulated 1 through 32 different species sown into a plot and then monitored the productivity over the long term. And this, over time, the differences in productivity became huge, from 20 to 180 grams per meter square. And that the magnitude of that difference is in blue here. And then that's compared to other manipulations of the same system in Minnesota of fertilization, manipulation of water, manipulation of CO2, herbivory fire. And what you can see is that the magnitude of the plant diversity effect is as big as other anthropogenic impacts on terrestrial systems. And so the loss of plant biodiversity then can rival other anthropogenic drivers of ecosystem change. And these things are not going to be independent. They're likely to interact. And the challenge in understanding that is we don't really understand what drives this productivity increase with diversity. So why are diverse communities more productive? And if we had a mechanistic understanding of that, then we would be able to forecast or think about mediation of the joint effects of potential biodiversity, plant biodiversity loss, with other factors such as climate change. So this talk, I'm going to talk about what determines the productivity benefits of plant biodiversity. And ecologists have long felt that the factors that drive the productivity benefits of biodiversity, or overyielding, will be the same factors that drive the stability in coexistence of these competing plant species. So the question of what maintains diversity of plant communities is thought to have the same answer as what determines the productivity benefits of plant diversity. And for years, the major explanation for both of these questions has been resource partitioning. So we can illustrate resource partitioning here, where we have two axes of resources. In the blue, species occupies or does well with one particular collection of combinations of those resources, and the yellow species does well with a different combination of resources. And the limited overlap in the resource in which they both do well reduces the intensity of interspecific competition, which can explain why those two species might coexist. And when you have plots that include those two species, they're more fully able to utilize the resources available, so that then could generate this productivity response. So then resource partitioning can do it all. It can explain coexistence. It can explain overyielding. And the challenge has been in these biodiversity manipulation experiments, it's been very challenging to demonstrate resource partitioning. So there's many studies which have been frustrated in demonstrating this mechanism. So this could be that resource partitioning is real, and we just are inadequate as humans and to actually measure the resources that matter to plants. And that I think is likely a big part of this, or in the traits that actually govern the resource dynamics. But it is also possible that other mechanisms are important, such as pathogen dilution or abiotic stress alleviation. And so if we go back to the evidence for resource partitioning, a lot of work has been done on the tallgrass prairie system to test competition and resource partitioning by David Tillman. And through the major interpretation of his work is that the tall grasses, such as big bluestem, are competitively superior to the other species. And his work suggests that these species should exclude them. Instead, this is what a stable prairie looks like. And so different from a dominance of tall grasses. But if you take seeds from this prairie and put it into a nearby post-agricultural soil, then you get the exact prediction of David Tillman's work, a dominance of the tall grasses, a lower diversity community that's dominated by the native tall grasses. So we found that if we take seed from this prairie remnant along with the microbiome and put it in the post-agricultural soil, then you can increase the diversity of that restoration and particularly increase the representation of a lot of the species that are common in the prairie remnants. So we're going to ask whether plant microbe interactions can contribute to coexistence and overyielding, and explaining some of these patterns that we've been exploring. So in this talk, I'm first going to present the framework that Andrew so generously talked about as an approach to evaluating the importance of microbial dynamics on plant-plant interactions. And then I'm going to summarize many studies that have used this framework that suggest that pathogens may be important in driving plant community structure. And then I'm going to present tests of the patterns of plant pathogen dynamics and diversity, and test whether pathogen dilution can drive overyielding. And then we're going to return to resource partitioning in the same system that we tested the pathogen dilution to test whether resource partitioning also contributes to overyielding. So just to orient you, plants interact with lots and lots of microbes. These are oospores of oomycetes, water molds are common plant pathogens. These are fungal pathogens in this cleared necrotic root. These are bacteria infecting the root tip, so lots of things that can decrease plant growth. Then there are buscomycorrhizal fungi, nitrogen-fixing bacteria that can play major roles improving plant growth, improving their access to resources. All of these could be important determinants of dynamics of plants. And there's tens of thousands of other microbes that plants are also simultaneously interacting with. And so the challenge of understanding how important these microbes are is big. And so in my dissertation work, I developed an approach where if we think of one plant and its microbiome, and first think of how that microbiome changes in response to that plant, and then how the change in the microbiome alters that plant's fitness. And then it could also alter the fitness of its competitor, which then could feed back on that plant. And the competitor is changing the microbiome, which has direct and indirect feedbacks. If you represent all of this mathematically, you find that the dynamics are dependent on an interaction coefficient, which is the difference of these direct and the indirect feedbacks. So if they do better with their neighbor's microbiome than their own, that'll generate a negative feedback, and that's the condition that'll drive coexistence. If they do better on their own, that'll generate positive feedback in alternative stable states. So the advantage of this result is that we can test this stability condition, and we can do it by taking one microbiome, put it in replicate pots, allowing the microbiome to differentiate on the host. And then we use these differentiated microbiomes in a test experiment. We grow the two plant types in the two different microbiomes. And then we evaluate whether they grow better in their own versus each other's microbiome. And that corresponds to the stability condition. So if they grow better in each other's than their own, then that is the condition for coexistence. So I did this in my dissertation work, and then it's been done a lot since then, as Andrew suggested. And this is a meta-analysis that we did about 10 years ago, so it's not complete. But with this meta-analysis, we can generalize what the importance of the soil microbiome dynamic. And so here we're looking across 920 studies that had used whole soil, whole plant interacting with the whole soil microbiome, and we generally find negative feedback. If we exclude the microbiome except mycorrhizal fungi, it's weaker. If we exclude mycorrhizal fungi, it's more negative. So that's consistent with plant pathogens playing the major role in driving the negative feedbacks. We also find that if the two plants that we're comparing are co-evolved with their microbiome, they're both native and they're interacting with native microbiome, then we have strong negative feedback. If one's not native, one's native, we have weaker negative feedback. If they're both not native, so a naive interaction with the microbiome, we don't have consistent feedbacks with the microbiome. So this is consistent with the microbiome dynamic contributing to coexistence of native plants, and it's consistent with escape from host-specific pathogens contributing to the success of non-native plant species. And the other result that we found was that when we compare the phylogenetic distance between two plant species, if they are phylogenetic very different, so from different plant families, then we have stronger negative feedbacks. If they're from the same genus or different genotypes of the same species, we have much weaker negative feedback. So this phylogenetic structure will contribute to phylogenetically diverse plant communities. Phylogenetically similar species are less likely to coexist. So the way to think about this is plants defend themselves against pathogens. All plants have different defensive systems. It's costly for the pathogen to overcome that defense, and then that drives specialization. And once you have specialization, then whenever any one plant builds up to higher densities, the pathogen on that plant accumulates, and that reduces the density of that plant. And so this process can lead to coexistence of many plant species in a community. So if host-specific pathogens drive coexistence, then can the dynamics of pathogens contribute to patterns of diversity, and can it contribute to the benefits of diversity? And tomorrow we'll talk about succession and restoration with mycorrhizal fungi. So I'm going to report on a project that was funded by the US National Science Foundation and the China National Science Foundation through a Dimensions of Biodiversity program. And we were looking specifically at the precipitation gradient between central Kansas and Illinois. And so what we find is as the precipitation increases, the diversity of prairie remnants in these solid dots increases. This is just the plant species richness in prairie remnants along that gradient. In the open dots is the post-agricultural grasslands, and they don't have a significant relationship, and they have much lower richness overall. But there's a similar gradient in China, in the rain shadow of the Himalayas. There's low rain, and then as you go east again, there's moisture coming in, and so we have from low to high precipitation. And as you go along that, plant species richness again increases with precipitation. So higher precipitation might be conducive to greater pathogen impacts. Many pathogens are able to infect. Their conditions in which they infect are facilitated by high moisture. Some of the pathogens I showed you, the oomycetes, they're water molds. They need high moisture to complete their life cycle. So there's reason to a priori expect that higher moisture conditions will be more conducive to stronger pathogen effects. And so we're going to ask whether precipitation drives stronger plant pathogen feedbacks, which then should generate the plant diversity pattern that we see here, but also a parallel pattern with pathogen diversity. And then does precipitation and pathogens drive increases in overyielding? So here's the objectives of this grant, and I don't usually show people this, but we actually did all these things. And so I'm really, peculiarly proud of this. But the first two objectives is just testing the patterns of plant pathogen interactions and diversity. And the second two objectives are manipulative tests to see whether the patterns that we're seeing are causal. So first result is, as we already showed, the plant diversity increases with precipitation. At each of these sites, we sampled the soil and we sequenced out the pathogens, fungal pathogens and oomycete pathogens, and looked at the diversity of pathogens along that gradient. And we found, this is the result for fungal pathogens, that fungal pathogen diversity does increase with precipitation in the prairie remnants, but not in the disturbed old fields. So that's true. That was true for the fungal and the oomycete pathogens. So we have a parallel pattern here: higher richness, higher diversity in high precipitation. Is that a spurious association? Is that an accident, or does precipitation drive increases in diversity? So we tested that by having plots with high precipitation, low precipitation, ambient precipitation, where all plots had received about 80 different plant species, and then we just let it go and we monitored the dynamics that came out of it. We monitored just diversity once a year. After 3 years, there were differences, and I'm showing you here the differences after 7 years. And so we have an increase in plant species richness with high precipitation treatment. This is 50% above ambient, and low is 50% below ambient. And so with increasing precipitation, we were able to reproduce the environmental pattern that we see, suggesting that precipitation does cause the changes in richness that we observed. So do we have stronger plant pathogen interactions? To evaluate that, we sampled populations of this common prairie species, Silphium integrifolium. It's being domesticated as an oilseed crop in the Land Institute, which is located right here. And this is the western edge of the distribution. So we sampled 4 populations at the western edge, or the dry edge of the distribution, 4 in the middle, and then 4 in the wetter part of the distribution. And then Yaniv Brandwein at the University of Minnesota and his postdoc, Kyle Keepers, baited out resistance alleles and found that the diversity of resistance alleles increases with precipitation. Because carrying resistance allele is costly, then this is consistent with a legacy of stronger host pathogen coevolution with high precipitation. So we also grew all 12 populations in common gardens in Illinois and in the Kansas locations. And then Catherine Turner at the Land Institute and Eric Cassetta monitored foliar pathogens at all the common gardens, and then found that the western populations generally had higher disease symptoms compared to the eastern populations. So this again is consistent with eastern populations having a legacy of greater pathogen effects. So we've found greater plant diversity with precipitation, evidence of stronger ecoevolutionary feedback, and higher pathogen diversity. Do we see greater over-yielding and with high precipitation, and is it due to pathogens? So the idea that disease would decline with biotic diversity has been something that was put forward by ecologists studying Lyme disease in Connecticut. It ended up being very controversial. It's something that you might have heard about, this controversy during the COVID period. COVID resulted from biodiversity of disease spreading from a biodiverse source to humans, causing a lot of problems. So an illustration of the potential problem of this framework. But more generally, this expectation was justified based on trying to use a modeling framework that represents a single population interacting with a single pathogen. And so the basic assumption of this framework is that disease incidence will increase with the susceptible hosts with the density of susceptible hosts within that population. So there's no axis for consideration of multiple species of hosts. And so then to go from this framework to this expectation requires verbal models. And two different ecologists will have different verbal models, which makes this controversial. And then to go from this pattern to the pattern of productivity increasing with richness, again, takes more assumptions. So with the feedback framework, when pathogen specialization drives negative feedback, then the basic assumption, the basic driver of the whole dynamic is that the impact of those pathogens on the fitness of that plant will increase as the proportion of that species increases in the community. And then when that's true, because the average proportion of a species will decline with 1 over S, then the pathogen impacts will decline with increasing species richness of hosts. And because we're measuring species pathogen impacts, then the productivity by inference will increase with increasing richness. So this would be the expectation from the theory, and we're going to use this framework to test whether pathogen dilution drives resource productivity responses. And so we do it in this system. We're in the KU field station. We're factorially manipulate precipitation, plant species richness, and plant composition. So we're working with 6 different grasses, 6 different asters, 6 different legumes. So we grow them in monoculture, in replicated monocultures, and we grow them in polycultures of 2, 3, and 6 species where we choose all of the species from the same family. And we grow them with 2, 3, and 6 species where we choose species from different families. And we did this keeping the overall representation of each of these species constant across all of these treatment combinations. And so we started this experiment by bringing in native prairie microbes. There was a prairie remnant south of Lawrence that was being destroyed by road expansion. So we were able to get truckloads of prairie soil, which we used to inoculate this post-agricultural site where we're doing this study. And we tilled in about 2 inches of prairie soil into every plot, 2 inches depth. We also started the experiment with plugs of each of the species that was inoculated with prairie microbiome. And then this is the Silphium integrifolium monoculture plot established during that first growing season. At the end of that growing season, we took sample cords from every plot, and I'm going to particularly talk about the monocultures. So we sequenced the microbiome from every monoculture, and we're particularly interested in whether the composition of the pathogen community is different between the different plant species. So during one growing season, did the composition of the microbiome differentiate, and particularly did the pathogen composition differentiate? And we used the monoculture soils in a test experiment to evaluate the feedback, how they grow with their own and other microbiome. And then we test whether pathogen dissimilarity predicts that feedback, and then whether the predicted feedback explains the overyielding in the polycultures in the next growing season. So we found that the difference in composition of the pathogens within one growing season. We found negative microbiome feedback, and we see that the more dissimilar the microbiome, the pathogen composition, dissimilar is this way, the stronger the negative feedback. We also found over-yielding in the next growing season. Sorry, this thing somehow got automated and I can never get it unautomated, so I have to wait for it. So it's this, and then when we combine plants that are very, that have, sorry about that. When we combine plants that have strong negative feedbacks, then we get more over-yielding. So, I mean, this relationship is significant because we have a ton of power. We have 240 plots. But you can see there's a lot of noise over here. There's less noise over here. And I'm going to return to that shape here with the next exercise. So we're parameterizing this predicted plant soil feedback is parameterizing from the differences in soil pathogen and oomycete pathogen. So we have that here, the dissimilarity of all pairs of fungal pathogens and the dissimilarity of all the oomycete pathogens. This is the best formula predicting feedback. So we can then represent a multi-species model of all 18 by 18 species driven by this pathogen dissimilarity. We can then determine whether different combinations of species, we tested every possible combination of communities, determine whether they would coexist mathematically, and then we can determine the productivity response for that combination just during the same way that we did for the over-yielding that we had showed you previously. So this is the result. We can predict from the pathogen differentiation the first growing season, we can explain coexistence of communities up to 9 species diverse. And then here we're looking at the over-yielding effect. This called complementarity. And the greater the richness, the greater the over-yielding. That is consistent with the results in year 2. And on this, here we're showing, this is the average mycorrhizal, negative feedback produced from the plants in that species pair and the average over-yielding from that. What you can see is that it's reproduced a significant relationship, but there is more noise at this end of the distribution than this end of the distribution. And you can see why, because this is the 2 species, 3 species, 4 species, 5 species, 6, 7, 8, 9. So as you go up in species, numbers of species in the community, it goes up in this stair-step fashion, which then creates more noise at this end when you combine it all. And we can test statistically that these 2 overall relationships are similar. So the noise, the shape of the noise here is similar to the shape of the noise here. So that gives added confidence that this process is important in this system. Okay, that was the first growing season. We are just finished the 8th growing season. We have 1 through 6 analyzed. And so I'm going to show you the patterns through the first 6 years. And I'm not going to talk about feedbacks anymore. I'm going to talk about pathogen dissimilarity in monoculture, because the logic is straightforward, right? So the red and the blue species, if we just have multiple axes of pathogen dissimilarity in monoculture, the red and the blue species share a lot of pathogens. So when you combine them, they're not going to get much release. But the red and the yellow species share fewer pathogens. So when you combine those, they're not going to have much overlap. So they're going to get more benefit from being near their neighbor because of the fewer shared pathogens. And that should translate to greater over-yielding, increasing with pathogen dissimilarity, which is what we showed in that first one. And we're just going to use this as the test of the pathogen dilution. Okay, so this is the years 1, 2, 3, 4, 5, 6. So productivity in polycultures is continuing to increase. These are long-lived perennial plants that put a lot of their resources below ground. So the next growing season, they're adding on biomass in that growing season. They're also reallocating some of last year's productivity to this year's biomass. But in monoculture, the rise in biomass is much reduced. If we relativize the productivity by the monoculture, this is the total over-yielding. Previous studies of this type have shown that The major driver of the long-term consequences of these biodiversity manipulations is through this statistical component called the complementarity. That's where all of the plants get bigger together. And that's what we expect to happen with pathogen dilution. Everybody benefits from dilution of their specialist pathogens. They all rise together. And so this has been the measure that we've been focused on and will continue to focus on. So here's that result. So this is release from fungal pathogens. This is dissimilarity of fungal pathogens, dissimilarity of oomycete pathogens in year 1, 2, 3, 4, 5, 6. So it increases, the strength of the relationship increases over time, and it's true for both of these types of pathogens. So the strength of soil pathogen dilution driving complementarity increases over time. So this experiment, we've been manipulating high and low precipitation. And when we look at that, we see higher complementarity and higher net overyielding with high precipitation compared to low precipitation. And this increase in overyielding is due to a decline in biomass in monoculture with high precipitation, and an increase in biomass in polyculture with high precipitation. So this decline here is difficult to explain. These are, yeah, we did add 50% more water than we got on rain during the growing season, but they're never flooded. It's still by, the middle of summer, there's big cracks in all of the soil. It's not like this is not waterlogged. And you could visually see a lot of, in the monocultures, some melted plants due to the foliar pathogens just wiping them out. So this is consistent with stronger pathogen impacts with high precipitation. So we have accumulation of host-specific pathogens suppressing yield in monoculture, dilution of those pathogens generating overyield in polyculture. We see that these effects increase over time, and they increase with high precipitation. And in independent trials where we manipulate the microbiome, we can show that including co-evolved pathogens increases the overyielding, sort of confirming this mechanism. And this is all this root pathogens. But all of the plants had foliar pathogens. And we started, Haley Burrow and Audrey Nelson started analyzing the foliar pathogens. I'm going to show you 3 years of data on the foliar pathogen. So looking across all the plant species and 3 years of data, we have an overall decline in disease incidence with increasing richness. We also had a decline in disease incidence with increasing family richness, because remember, we grew them for this given level of diversity, we have them with same family neighbors or different family neighbors. So this is consistent with the foliar pathogens of within the same family, more likely to share pathogens. So these are getting less benefit from dilution. So foliar pathogens are diluted by both species and family richness. And so we can take the amount of that reduction in foliar disease for each of the species and then come up with an estimate of how much release they would get in polyculture from that combination of species. And that release, the predicted release from foliar disease, predicts complementarity in the 3 years that we had measured it. So foliar diseases are not all of the symptoms, or just the symptoms, but not the entire effect of potential foliar pathogens. And so we sequence the foliar microbiome, particularly foliar fungi, and we could pull out the pathogens, or we could look at everything that wasn't a pathogen, and we again find that the dissimilarity in monoculture predicts the overyielding. And in fact, this relationship was stronger in many ways, in several cases, than the actual disease incidence, which is consistent with the disease symptoms underrepresenting the net impacts on plant growth rate and plant fitness. So we can add on here that dilution of both foliar and soil pathogens generates overyielding. So the way to think about this is, if you have a favorite plant that you plant out a lot of it because it's pretty, black-eyed Susan, then disease of black-eyed Susan spread from themselves to their neighbor and back again. And with that amplification of disease, you end up having reduced success, reduced growth of the black-eyed Susan. If black-eyed Susan was in a diverse mixture, there might still be diseases on black-eyed Susan, but the overall performance of that individual would be greater, and along with the total productivity. So we see then that we have evidence that specialist pathogens can drive coexistence, diversity patterns, and the productivity responses to plant diversity. So what about resource partitioning? We started out talking about the importance of resource partitioning. So if we go back to this representation, this should provide a way to test it, right? So the blue and the red species, if these are two axes of resource utilization, then they have greater overlap in resource utilization. They should have less release in combination compared to the red and the yellow species. So that same approach, the dissimilarity in resource uptake, should also predict overyielding. So the challenge that we had, and the challenge of plant ecology, is measuring resource appropriately. And so we weren't able to do that. We did measure lots of things, but I never was confident that it was the right thing. But we did take the approach that has been commonly used, which is to measure traits. And so we can group those traits. This is part of the dissertation work of Laura Podzakowski. Laura grouped these traits by physiochemical, root traits, or shoot traits. And then we can look at whether that predicts overyielding, and it does. So again, increasing over time, and each of them does, along with the temporal dissimilarity, the seasonality also did that. So for these traits, though, they're not independent. And when Laura directly evaluated the non-independence, she found that this was the biggest driver of this relationship. This was the stronger association, the physiochemical dissimilarity. So we can then look at the joint effects of physiochemical trait dissimilarity and fungal pathogen dilution. And together we are explaining more of the productivity response to plant diversity than we're able to explain with either of them alone. So that is consistent with multiple mechanisms contributing to overyielding and coexistence. So just to be clear on what we have, the evidence, the degree of confidence we have in the evidence, what we have evidence of is that we see that changes in pathogen composition drives overyielding. We don't know what plant traits govern the host specialization that we see, the pathogen specialization. And then we have evidence that plant traits that likely relate to resource acquisition also generates overyielding. And we don't know what resources are driving this. This then is a bit more of an indirect measure compared to this. So we really want to know what resource is being partitioned. And we took an approach of just measuring the elemental composition of every plant. So we sampled the leaf tissue of every plant in every plot, and we did it over 2 years. And then we measured the concentration of 20 elements in Ivan Baxter's lab at Danforth Plant Science Center in St. Louis. And overall, the dissimilarity in elemental composition, again, predicted overyielding. So that's awesome. We have 20 elements. Probably some of them are irrelevant. So we just did a little bit of a fishing exercise to figure out what is the best combination of elements that explains overyielding. And that's what this is. So magnesium, potassium, cobalt, nickel, and cadmium. So I don't know a lot about plant chemistry, but for those of you who do, are these the ones that you would have picked out as being the most important elements? So it's possible that these are the resources that plants are competing for. It's also possible these are proxies for soil depth. And so that's something that we're following up. We're measuring the concentration of these elements with depth in these soils. But it's also possible these are proxies for differences in plant defense. And that's something that we can't rule out either. So overall, this goal of what's funded, this project is currently funded by the Biology Integration Institute, which focuses on perennial plant traits. And the goal is to understand how plant traits mediate what the community productivity effects, which I've showed you some of that, but also how it then translates into ecosystem properties, and particularly soil carbon. After 5 years now, we have seen increases in soil carbon with plant species richness. And again, we have a ton of power. We have 240 plots. So, the actual magnitude of increase isn't that great, but it's still a significant effect. And that's after 5 years. We're still processing the samples taken after 7 years. And then after next year, we'll take samples again. We expect that these effects will become stronger over time, and that this experiment, by having lots of different combinations of plant traits, could be a good platform to try to evaluate which combination of plant traits govern the increase in soil carbon and soil recovery. But so overall, we have evidence that the restoration of plant diversity and microbial composition does provide a path towards restoring ecosystem functions, which then can give hope that we might be able to reverse the decades of degradation done by conventional agriculture. And I will want to thank a lot of people. But for the work that I presented here, Peggy Schultz has been helping me manage all of this experiment, and I presented work by Laura Podzakowski, Haley Burrow, Guanzo Wang, and Martin Ipinga did the theory. But those are people who've been analyzing the data. There are so many people that helped manage this study, and so many collaborators in different institutions. And so I'm grateful for all of them. So I'm grateful for you for being patient with me and as I talked about this work. And so I'm happy to take any questions.
Wondering if you had that slide with the 3 graphs for microbiome prediction pathogens driving diversity.
Yes.
High richness is the x-axis.
Yes.
And I was wondering if it seems like evenness might have a big effect as well.
Yes, I think you're right. And in this study, we just manipulated richness with keeping, when we started the plants, they were all hyper-dispersed. So they were very evenly distributed throughout and so we sort of are maximizing the interspecific interactions that might exist from the beginning. So that could explain why we were able to see productivity responses so quickly. But you're right that evenness, if we kept richness constant and manipulate evenness, I agree with you, that would also be important. Yeah.
I jotted down just that you looked at dilution in the foliar pathogens being predicted by species and family richness, but I wasn't looking up, and I was wondering, was the relationship stronger for family richness than species, or can you infer that foliar pathogens are then like less specialized or more specialized with that relationship than other pathogens?
That is a good question. We statistically, they were about equal, but that is not a good representation of the effect size. So, but when you compare it to the other pathogen, the other microbes, all that soil, I didn't mention the family at all with the soil story. And that is because the it actually was just not important at all, and surprisingly so. I just want to, you can see it in this figure. Because I'll just, so what you're seeing here, it's organized by family. This is the dissimilarity amongst asters. And we had stronger dissimilarity with stronger negative feedbacks amongst asters than this is the dissimilarity amongst legumes. So we had very weak dissimilarity for oomycetes and fungal pathogens amongst legumes, and we had positive feedbacks amongst legumes. So we have this, so we didn't see that bigger differences. You know, we would expect a checkerboard pattern when we look at these between family comparisons here, and we don't see it. So we didn't see a difference between, and when we go on to this effect here, I didn't show you the family effect because it ended up not being significant. So we are seeing as strong an over-yielding response within family as between family. I think that over time that's been fading. There was an effect that early on, but that's been I think that that family effect might be increasing, I'm sorry, in this system. At least in the first 6 years, we didn't see a family differences. So what we're seeing with the foliar pathogen wasn't realized in the total data or in the soil pathogen data. So it was stronger. The family effects was stronger with the foliar pathogen than what we were seeing in the other parts of the data.
Yes, I've got a question going more so like back to the beginning, more so like with your focus for you.
Yeah.
It's like what Andrew said at the beginning, like you're one of the fathers of like soil feedbacks. So I was curious to hear about really like how you got into that and how you were able to tap into an area of science that people didn't even know existed.
Well, I wanted to look at coevolution of plants and mycorrhizal fungi, and I didn't have anybody around who knew how to work with mycorrhizal fungi. And so I designed an experiment that I thought would be the right one to do, which is train them and test them. And I wanted to think about mycorrhizal fungi. And in the end, I did the experiment and they did worse in their own soils. And I could not convince myself that it was because of mycorrhizal fungi. And I started looking at these pathogens and it was obviously because of pathogens. And so then I had a problem because I had a modeling framework to think about coevolution of mycorrhizal fungi, plants and mycorrhizal fungi. And now I had this experimental approach that was not about that. And then I had to develop a theoretical a mathematical framework that matched the experiment that I actually did. And so I sort of just backed into it. And it was, yeah, the modeling framework that I started with, which is in my dissertation, wasn't the one that I showed you. But it has the same assumptions. And I've just sort of recently, when I was trying to apply this model to animal host microbiome interactions, did the work of actually putting those two modeling frameworks together. And is that because I needed to, because they don't have the right experiments with animals. They don't allow you to train microbiomes on an animal and test it. So So we had to imagine we had to start with non-random association with the microbiome and get proxies for it. Anyway, so I did just recently return to those two different things yeah.
So I do have a question, and I always like to preface. I'm a fish guy. So I don't sound like I got all that topics because I don't. But I have a question regarding wetlands, and this is going to take a little bit of extrapolation from this. But wetlands tend to be monocultures, right? They tend to deviate from that for a variety of reasons. And so extrapolating with this data, we know that the pathogens tend to occur more wetter environments. And so when these results here, I would suspect that you would have just overwhelming disease issues at wetlands that would really inhibit the production of monocultures. Is there kind of like a critical threshold where the pathogens, either since wetlands typically are more wet, is there like some sort of pathogen nature of
Yeah.
change?
Yeah, I think that's a good point. But I will point out that like the whole state of Illinois was originally wetland. It's the whole state's been what's it called when you put in been tiled to drain it so they actually, and so it wasn't always wet though, right? It had seasons where it was wet, seasons when it was probably would have been inundated, and then it would dry out in the summer and it would become very dry in the middle of the summer. And so like that kind of wetland is like Illinois shows can be highly diverse. And if you have something that's perpetually wet, then you get this anoxic, continually anoxic soil, then that is different. And so I think that there are, so I think or salt marsh type situations similarly, where you have these, where the environment ends up imposing a stress, which is not just a stress for the plant, but also a stress for potential pathogens of that plant. Then you can get these more stable, lower diversity systems. That's my suggestion. So
Yeah, I think that actually makes a lot of sense with anoxia
yeah,
gens up to a point once anoxia kicks in, they're not able to persist there.
Yeah.
And then you get back into whatever plants the most fit is now dominating.
Yeah.
That makes a lot of sense.
Yeah.
I was looking how beneficial microbes fit into this framework of like you have more diversity, so dilution of pathogens, but where do you think the beneficial microbes?
It's a really good question. So tomorrow I'm going to talk about plant mycorrhizal fungal interactions, but I'm going to talk about it in the context of turnover, positive feedbacks and turnover during succession. And it does fit into this story, and not in this study. It was a parallel study, which we manipulated the initial microbiome composition. And in that study, we created a bigger range of microbiome compositions, including the whole soil, but also only prairie mycorrhizal fungi, and then the degraded microbiomes, the 3 treatments. But in that, we did include, we do see differences in mycorrhizal composition in monocultures. And when we include that as a predictor, the work I'm going to show you tomorrow shows that we have positive feedback through mycorrhizae in this system. And then we get that same result where with positive feedback, you'd expect that the more dissimilar you are, the stronger the positive feedback, the lower the over-yielding. And that's what we were seeing. We were seeing a negative relationship between mycorrhizal dissimilarity and over-yielding. But we also saw an interaction in that experiment. We manipulated mycorrhizae and pathogens somewhat, not completely independently, but we did see an interaction of mycorrhizal dissimilarity and pathogen dissimilarity, such that when we had different mycorrhizal composition, it did inhibit, it didn't reverse the relationship between pathogen dissimilarity and overyielding, but it lowered it. And so there was a significant interaction between the AM fungal dissimilarity in monoculture and the pathogen dissimilarity in monoculture in determining the level of overyielding. So mycorrhizae are important in this system, and they work against the pattern of overyielding that we're seeing. So.
Right, that's our time, but you can, welcome to stay around. We'll be in the lobby, you can talk more. Thanks.
So, yeah. Thank
Good
you,
job.
Andrew. It was really
Great.